Hello students, today we will discuss about pyrimidine and purine metabolism. So let's come to the introduction. As we know, human tissues can synthesize certain metabolic compounds, and among them purine and pyrimidine is also one. It is mainly synthesized in the cells of liver. Their major role is to synthesize nucleotides, which is the building block of RNA and DNA, consisting of a nitrogen-containing base, a pentose sugar, and 1 to 3 phosphate groups. So let's come to the structures of purine and pyrimidine. Pyrimidines are 6-membered heterocyclic aromatic rings containing 2 nitrogen atoms at position 1 and 3. They form the nucleus of uracil, thymine, and cytosine bases of nucleic acids. And the purine ring structure is represented by the combination of a pyrimidine ring with a 5-membered imidazole ring to yield a fused ring system containing 4 nitrogen atoms at position 1, 3, 7, and 9. They are the main structure of adenine and guanine bases. Role of purine and pyrimidine. They act as substrate for nucleic acids, RNA and DNA, biosynthesis, and in the repairing system. It is the components of many coenzymes such as nicotinamide adenine dinucleotide, flavin adenine dinucleotide, coenzyme A, which are involved in various metabolic reactions. They also act as effectors molecules such as AMP, adenosine monophosphate, ADP, adenosine diphosphate, ATP, adenosine triphosphate, GTP, guanosine triphosphate of some allosteric enzymes to regulate the metabolic reaction. Now let's come to the biosynthesis of nucleotide. They are synthesized by 2 different types of pathways, and both these pathways are equally important in cellular metabolism. Number 1. De novo pathways. In this pathway, the nucleotide bases are assembled first from the simpler compounds such as amino acids, ribose 5-phosphate, carbon dioxide, and ammonia. Number 2. Salvage pathways. In this pathway, the free bases and the nucleosides obtained from the degradation of nucleic acids are recovered and reattached to the ribose unit to get nucleotide bases. So let's see de novo purine nucleotide biosynthesis pathway. This pathway can be divided into 2 phases, phase 1 and phase 2. The steps involved in the phase 1 biosynthesis are as follows. Number 1. The first committed reaction is the formation of phosphoribosyl pyrophosphate, in short PRPP, from ribose 5-phosphate catalyzed by the enzyme PRPP synthetase in presence of ATP. Then, the amino group of glutamine displaces the pyrophosphate from PRPP, yielding 5-phospho-beta-D-ribosylamine. This reaction is catalyzed by glutamine PRPP amidotransferase. 5-phospho-beta-D-ribosylamine then reacts with the carboxyl group of glycine to form glycinamide ribonucleotide, which is catalyzed by the enzyme glycinamide ribonucleotide synthetase. It is an ATP-dependent reaction where high-energy bond is utilized for this condensation process. Number 3. Glycinamide ribonucleotide is formilated by glycinamide ribonucleotide transformylase to give formyl glycinamide ribonucleotide. Number 4. The carbonyl oxygen of amide is replaced with amino group of glutamine to form formyl glycinamide ribonucleotide, in short FGAM, catalyzed by an ATP-dependent formylglycinamide ribonucleotide amidotransferase enzyme. Number 5. The enzyme cyclase or 5-aminoimidazole ribonucleotide, in short AIR, synthetase catalyzes the formation of 5-membered imidazole ring in an intramolecular ATP-dependent reaction. By eliminating water molecule between the amide group and N-formyl group, the product form is known as 5-aminoimidazole ribonucleotide, in short AIR. Number 6. In order to complete the second ring structure, the first reaction is the addition of carboxyl group on the amino group yielding N5-carboxyaminoimidazole ribonucleotide catalyzed by an ATP-dependent carboxyaminoimidazole ribonucleotide synthetase. And in number 7, the position of carboxyl group is rearranged by transferring onto carbon number 4 of imidazole ring, and this step is catalyzed by the mutase enzyme to form carboxyaminoimidazole ribonucleotide, in short CAIR. In step 8, this step involves the formation of amide bond between the carboxyl group and the amino group of aspartate in presence of ATP to form N-succinyl 5-aminoimidazole 4-carboxide ribonucleotide, in short SAICAR, catalyzed by the enzyme SAICAR synthetase. In step 9, in this reaction, the enzyme lyase helps in eliminating the carbon skeleton of aspartate as fumarate, giving 5-aminoimidazole 4-carboxamide ribonucleotide, in short AICAR, as product. In step 10, in order to complete the whole structure, the final carbon atom is contributed by formyl tetrahydrofolate to 5-amino group catalyzed by ICAR transformylase, and the product form is known as N-formylaminoimidazole 4-carboxamide ribonucleotide, in short FAICAR. In step 11, a 6-member ring is formed by eliminating water molecule between formyl group and carboxamide group to form inosine monophosphate, in short IMP, which is the main precursor of other purine bases. This reaction is catalyzed by IMP synthase. Next, phase 2 reaction sequence. The inosine monophosphate IMP formed in the last step of phase 1 sequence is then converted to adenosine monophosphate, in short AMP, and guanosine monophosphate, in short GMP, in two pathways. Number 1. Conversion of IMP to AMP. In this step, first, aspartate condenses with IMP to give AMP, which is a GTP-dependent reaction, catalyzed by adenylosuccinate synthetase to form adenylosuccinate. In the second step, adenylosuccinate lyase releases fumarate from adenylosuccinate to give AMP. This is how inosine monophosphate is converted to adenosine monophosphate. Number 2. Conversion of IMP to GMP. This conversion is also completed in two steps. Firstly, IMP is converted into xanthosine monophosphate, in short XMP. with the help of IMP dehydrogenase and NAD+, which is followed by addition of amino group derived from glutamine in presence of ATP-dependent XMP glutamine aminotransferase to give GMP. Next is regulation of purine nucleotide biosynthesis. The regulation of purine nucleotide biosynthesis occurs at two levels. Number 1, the purine nucleotide biosynthesis is mainly regulated by PRPP synthetase and glutamine PRPP aminotransferase. Number 2, another regulation occurs at the branching pathways leading to the formation of AMP and GMP from IMP. Adenosuccinate synthetase, IMP dehydrogenase are inhibited by ATP and GTP respectively. Now come to the de novo biosynthesis of pyrimidine. Reaction sequence involved are as follows. Number 1. In the first committed step, alpha amino group of aspartate reacts with the carbamoyl phosphate to form N-carbamoyl aspartate by the help of aspartate transcarbamylase. In the second step, pyrimidine ring formation occurs, which is catalyzed by the dihydroorotase with the elimination of water molecule, and the product formed is known as L-dihydroorotate. In the subsequent step, orotate is formed in the NAD+ dependent dehydrogenation reaction catalyzed by dehydrogenase. Once orotate is formed, the orotate phosphoribosyl transferase transfers ribose 5' phosphate side chain from PRPP to yield orotidylate. Orotidylate is then decarboxylated to uridine monophosphate, in short UMP, by decarboxylase, giving carbon dioxide as byproduct. UMP is again phosphorylated to yield uridine 5' triphosphate, in short UTP, in presence of kinase by consuming 2-ATP. UTP is then converted to cytidine 5' triphosphate, in short CTP, with the action of cytidylate synthetase by transferring amino group to glutamine with the consumption of 1 ATP molecule. Now, synthesis of thymidylate. We all know that DNA contains thymine rather than uracil, and the de novo pathway to thymine involves only deoxyribonucleotides. First, ribonucleotide reductase converts CDP or UDP into deoxy-CDP or deoxy-UDP. With the help of nucleoside diphosphate kinase, deoxy-CDP or deoxy-UDP is converted to deoxy-CTP or deoxy-UTP. Later, enzyme deoxy-UTPase catalyze the reaction of phosphate group removal from deoxy-UDP to give deoxy-UMP, and deoxy-UMP is converted to deoxy-TMP in presence of thymidylate synthase. Let's come to regulation of pyrimidine nucleotide synthesis. De novo pyrimidine biosynthesis is regulated by the enzyme carbamoyl phosphate synthase 2, aspartate transcarbamylase, and CTP synthase. Next is degradation pathway. Number 1. Degradation of purines. Purine nucleotides are degraded by a pathway in which they lose their phosphate through the action of 5' nucleotides to give AMP. AMP yields adenosine, which is again deaminated to inosine by adenosine deaminase. Then inosine is hydrolyzed to hypoxanthine, its purine base, and D-ribose. Hypoxanthine is oxidized successively to xanthine and then uric acid by the action of xanthine oxidase. Molecular oxygen is the electron acceptor in this complex reaction. So, the final product of the purine degradation is the uric acid in human. Next is degradation of pyrimidines. The degradation of pyrimidine nucleotides can proceed in 3 phases depending on the organisms. In first phase, it undergoes dephosphorylation, deamination, and glycosidic bond cleavage reactions to yield free bases. In second phase, uracil and thymine undergoes sequence of reduction, hydration, and deamination reaction to yield beta-alanine and beta-amino isobutyric acid, in short BAIB, which is excreted in urine. In third phase, beta-amino isobutyric acid and beta-alanine undergoes series of reaction to give manoline-CoA and ultimately converted to carbon dioxide. Next is the salvage pathways of purine biosynthesis. This pathway involves 2 enzymes, namely adenine phosphoribosyltransferase transferase, in short APRT, and hypoxanthine guanine phosphoribosyltransferase transferase, in short HGPRT. Both these enzymes utilize PRPP as a source of ribose 5' phosphate group. Adenine phosphoribosyltransferase transferase will catalyze the formation of AMP, whereas hypoxanthine guanine phosphoribosyltransferase transferase catalyzes the formation of GMP as well as IMP. Next is salvage pathways of pyrimidine biosynthesis. Similarly, with that of purine, pyrimidine also has salvage pathways, where free pyrimidine bases are salvaged to reform into functional nucleotides. For this, the necessary reactions are described as: ribose sugar is reattached to the monophosphate by the action pyrimidine phosphoribosyltransferase transferase and phosphorylated by its substrate-specific kinase, except thymidine, which require deoxyribose 5' phosphate and thymidine phosphorylase. Purine and pyrimidines are essential to carry out many life processes, since they constitute the building blocks of DNA and RNA. The de novo pathway and the salvage pathway equally important in the nucleotide metabolism. They seem to be nearly identical in all living organisms. By studying this metabolism, many scientists were able to develop therapeutical drugs for the diseases caused by the defective enzymes involved in the reaction of the pathways.